Thank you. Can you hear me? Yes. Good. Yeah, we have a lot of co-authors on this. I'm based in Finland, but we're kind of scattered across all of Europe. I guess I need to point that way. This is one of the work packages of an EU project on marine restoration, looking at marine restoration in 3 ecosystems, deep sea ecosystems, hard bottom coastal shores, and soft bottom coastal shores. So work package 2 is ours, shallow, soft bottom marine habitats, and we're focusing on seagrass. In brief, the whole goal of this is to improve these kind of horrible restoration success numbers that we have for seagrass. So we're developing strategies to do that. The nice thing about presenting last, most people have talked about this already. One of the most important things is to reverse habitat degradation before restoring. There's no point in restoring if we haven't dealt with the pressures. But what we're focusing on is this aspect down here, is understanding the role of interactions and feedbacks between ecosystem engineers in a restoration context. We know that these might be potentially very important for seagrass and also salt marsh restoration. And we're focusing on interactions between 2 groups of ecosystem engineers, plants and bivalves. So we have several different ways in which we're addressing this within the work package. We started with a systematic literature review. We did field surveys of seagrass beds across Europe, field experiments also across the European gradient, lab experiments both in still water and flume aquaria. So these 2 we started last summer, and I don't know actually why it goes 1, 2, 3, 1, 2. It should be 4 and 5. But these are ongoing. And then we have also a group in the Netherlands doing mathematical modeling to kind of understand the patterns that emerge. In addition to this, we have a network of case study sites, and those are existing and ongoing seagrass restoration in different areas using different methods. That's a lot. I'm not going to talk about everything, obviously, but my goal is to give you an overview of the lessons we've learned from the literature review and from the first year of the field and lab experiments and what that can teach us about restoration and that we can apply for the upcoming experiments. So we started with the systematic review and ended up with about 440 studies. For each study, we categorized it as positive, mixed, negative, or non-significant interaction. So positive and negative are pretty self-explanatory. A mixed interaction would be a study where there are both positive and negative effects occurring at the same time. So one example would be scallops in seagrass meadows have higher survival but lower growth rate than scallops outside seagrass meadows. So that's mixed because there's trade-offs going on. So the goal of the review was to see if we could find any pattern in the environmental conditions that determine the type of interactions. In other words, when do we get positive interactions, when do we get negative interactions, and can we use that in restoration context? We thought it might have something to do with stress. So in stressful habitats or conditions, there might be more facilitation. So we looked at habitat, intertidal versus subtidal, infaunal versus epifaunal bivalves, exposure. We also did exposure in the field experiments, and temperature. And there's also some spatial and temporal scale, but none of that was significant. Instead of using a traditional meta-analysis to look at effect sizes, we use cumulative link modeling, and that lets us put all of the studies into the analysis and determine which environmental factors contribute the most to the patterns or the probability of getting positive or negative interactions. And I'll show you an example of that coming up. So we did plants and bivalves across all marine and freshwater systems, but this review was dominated by subtidal seagrass habitats, but also intertidal seagrasses are quite well represented. And we get a nice mix of epifaunal and infaunal bivalves across most of the habitats. So the 2 big factors that came out of the analysis is intertidal versus subtidal and epifaunal versus infaunal. So in intertidal seagrasses, interactions are mixed. We saw the same pattern also in salt marshes, which was the other main intertidal habitat. And also that infaunal bivalves and intertidal seagrasses tend to be involved in more negative interactions than positive interactions. Whereas it's the opposite for epifaunal. And for epifaunal bivalves, there's more positive than negative. And we contrast that with subtidal seagrasses, where interactions are overwhelmingly positive. There are, of course, always both, but in subtidal seagrasses, plants and bivalves seem to facilitate each other. And also in the subtidal, where there's not really a difference between the epifaunal and infaunal bivalves. So to look at the differences here, we just looked at some of the mechanisms. So in intertidal seagrass meadows, epifaunal bivalves tend to provide sediment stabilization, whereas seagrasses tend to provide shelter from predation. In some cases, in other cases, they increase it. So we get a mix of both positive and negative mechanisms. So in bold, I don't know if you can see the bold, but generally the top ones are the most important mechanisms that we see the most. Whereas if we're looking at infaunal habitats, we get a lot of space competition in both ways, both seagrasses affecting bivalves and bivalves affecting seagrasses. So this tends to be the kind of dominant mechanism affecting infaunal bivalves in intertidal seagrasses and salt marshes. One important positive effect that we do have in with infaunal bivalves is sulfide reduction. This is done by a group of specialized bivalves, or two groups of specialized bivalves, the lucinids and the solimids. I'll get back to that. If we go into the subtidal systems, we get a lot more positive mechanisms on both sides, although we do again have some occasional negative trade-offs, like reduced food availability within a seagrass meadow. With infaunal bivalves, we again have mostly positive effects, but they are different mechanisms that involving infaunal and epifaunal bivalves, and that makes sense since they're above or below the sediment. Again here, sulfide reduction is really important, and that's a service provided by infaunal bivalves. The most important mechanisms here for bivalves is increasing light availability by filtering the water and enriching the sediment through the deposition of pseudofeces. And the other way around, seagrasses provide shelter and substrate for the bivalves. We also looked at things like temperature, and we found an increase in positive interactions with sea surface temperature. This was the other factor that came out significant in the analysis. Now this color scheme has shifted, sorry about that. So positive is this yellow or orange colored line. So the proportion or probability of positive interactions increases with summer sea surface temperature. And that's, again, might be linked to stress. So they are more stressed here. And this might, again, be linked to the sulfide-reducing bivalves, which tend to be found in the kind of warmer areas. So these are the sulfide-metabolizing bivalves, and we can compare them to the other infaunal bivalves and the effects they have. So they are much, much more positive than other infaunal bivalves. And the other thing that might be important for restoration, we compared invasive species and non-invasive species. So if you have a native bivalve and a native plant interacting with each other, you get, again, mostly positive interactions. But if you have a native and a non-native interacting with each other, you tend to get mostly negative interactions. This is especially true for invasive bivalves in native plants. The other way around is not quite as negative. But this might also be important for restoration. So after this, we also wanted to test this in the field in a variety of habitats within Europe. In the north, we have 4 sites using blue mussels in Zostra marina. And in the south, we have 3 sites using Pinna nobilis in either Posidonia or Cimodosia. And in the lab experiment, I'll talk mostly about the Mytilus zostra because that's what I was directly involved in. In the lab, we found that it could increase zostra growth, at least in an aquarium setting, probably through fertilization of the sediment. So I wanted to put this up here to show how we do research in non-tropical countries, because it seems most people here are working on tropical seagrasses. So this is me. We were setting up the site in Estonia, and it's about 1 degree on the boat and 3 degrees in the water. So that's fun. And this is what it looks like underwater. So this was a factorial field experiment. So we have plots with just Mytilus. We have plots with eelgrass. They're attached to a plastic frame and just buried under the sediment. We have plots with Mytilus and Zostra. And then we have controls in natural seagrass, if there's still a natural seagrass meadow at the site. A procedural control with just the plot, and then just bare sand. So sorry, this is kind of a not very nice title for a slide. But we also learn in these kind of experiments what goes wrong and how we can avoid things that go wrong. So in Finland, we had problems with filamentous algae. In the 3 northern sites, we had problems with loss of mussels. We had one of our sites in Finland had ice scouring. In the Netherlands, basically, they tried some stuff with seeds and seagrasses and everything was lost. And then in Estonia, we actually had the opposite problem where one of the sites was completely buried by sand. In the Mediterranean, things went a bit better, but in Turkey, there's grazing by invasive rabbitfish. And so that meant we had to put cages out to prevent that, but the cages are a bit of a problem in really stormy weather. So one of the sites was lost because all the cages were washed away and then everything was grazed. And of course, you were probably thinking this, but maybe using plastic meshes is not super sustainable if we want to do large-scale planting. But we also had some successes. So we had Zostra survival for the first year in all sites, at least a few shoots. And in about half the sites, we had expansion. Unfortunately, most of the mussels were lost, so we couldn't compare the mussels versus non-mussels. In the Mediterranean, we had very good results, except for that one Turkish site. So all the seagrass survived, all the pina survived, and seagrass promoted pina survival and growth, and that's really important because pinas are disappearing. And since the shell provides a lot of substrate, we also get higher associated biodiversity in plots that have both the bivalve and the seagrass. And based on all these results, we are planning new experiments in 2018 for the Zostra and mussel experiments. We're going to use biodegradable 3-dimensional substrates, so hopefully we get to keep the mussels and provide substrate for the seagrass and stabilize the sediment. So very quickly, I promised lessons for restoration. Co-restoration could increase both seagrass and bivalve survival and growth, as well as reestablishment of the associated community. And this is something that's often ignored in restoration studies. We focus on just the seagrass, but it's the whole community that we want to get back. From the review, we know that we should promote positive interactions. So that's generally most bivalves in subtidal habitats, epifaunal bivalves in intertidal habitats, sulfide-metabolising bivalves, especially in warmer climates, reduce negative interactions with infaunal bivalves in intertidal habitats, and reduce pressures prior to restoration. So that means dealing with invasive species, nutrients that lead to algal blooms, and also that we will have to stabilize sediments in areas where seagrass has been completely lost. And that's about it. Thank you very much.
Thanks, Karine, for a really interesting talk. Any questions for Karine?
Have you considered looking at whether seagrass improves reproduction among bivalves in areas that have experienced significant ocean acidification?
That would be interesting. We would just have to find sites where that would be the case. But that would definitely be one of the metrics that we could measure for bivalves, but that would have to be done on a much longer time scale, I think.
Any other questions?
Thank you. I'm glad your pina is alive.
Yes, it's doing really well in some of the sites.
I was wondering if you know that there is a net of monitoring for they are looking for alive individuals of pina. It will be great if you upload your observation because they are looking for resistant individuals to the parasite.
So where are they looking for it? Anywhere in the Mediterranean?
Yes, anywhere
Okay.
in the Mediterranean.
Yeah, so these experiments are in Croatia and Italy and Turkey, so Eastern Mediterranean.
That will be super great.
Okay, yeah, we should talk. Yes.
Any other questions?
There's been a lot of effort recently for oyster restoration. Did you have a look at any benefits for seagrass or oysters at that larger scale?
No, we haven't focused on oysters basically because we don't have it in the Baltic. So we can't do but in the Netherlands they could do it, and we have one student who's actually looking, he gave a talk, he's up there. He gave a talk about oysters and seed trapping of seagrass, but we're not specifically looking at oyster restoration.
So when you're looking at your interaction strengths, do you take into account anything to do with density? Because if you're looking at a result from one experiment and it has a positive interaction, but the densities are extremely different from another.
Yeah, no, we just categorize each study as one or the other, and that's one of the reasons why we decided not to go for the meta-analysis, because it was, if we had to deal with density dependence, we would only get a very small proportion of the studies that we could actually analyze in a meta-analysis, and that wouldn't be so useful for kind of global lessons that we could learn. But in terms of, there were studies that had density dependence, so at a low density, bivalves would facilitate seagrass, and at a high density, they would harm it, and that would go into a mixed interaction. So that's how we categorize those studies.
Hi. Yes, I've been working in the Baltic Sea on seagrass and mussels and their interactions. And I mean, they don't really mix. They do like, it's sort of mosaic where you have like a pack of mussel in the middle of seagrass, and you have a pack of mussel before, a pack of mussel after when there's not enough light. So, and there are reports that they are actually excluding each other, and they're even, in Denmark, they even calculated the amount of mussel. Yep. So, isn't it some sort of problem for this kind of project?
No, well, no. In our seagrass meadows, we do have patches like that, but we also do have mussels just kind of scattered throughout the seagrass. One of the questions with the modeling approach that the Dutch team is taking is looking at pattern formation of these kinds of things. But for the experimental work, we're looking at putting the mussels into the seagrass, because that's what we actually see in our meadows. So yeah, just mussels scattered around. In the review, the manuscript of the review, where we go into a lot more detail, we do deal with kind of cross-habitat versus within-habitat interactions. So that would be the difference between mussels scattered within a seagrass meadow versus a mussel reef and then a seagrass meadow. So we do actually deal with that within the review. I just didn't talk about it here.
Okay. Thanks very much, Karine. I'd just like to thank all of the speakers this afternoon. I think it's been a really good session and really well attended. Thank you all for coming, and I'll let you go and have coffee.